Hi, everybody. This is Alexa Tinsley, the Director of Community Engagement here at JAP Foundation. Thank you all for joining us today as we welcome Dyne Therapeutics to our community webinar series. We're going to get started in just another minute. We're going to let people hop on. So we will be right back. Thank you, guys. I'm going to share my screen. Can everybody see that? That look good?
That looks good. Can you hear me okay?
Yep.
Super.
All right. Awesome. So again, everybody, thank you guys so much for hopping on. I am Alexa Tinsley, the Director of Community Engagement here at JAP Foundation. We really appreciate you all joining us today. We are very excited to welcome Dyne and their team to discuss moving the bench to clinic to deliver a potentially transformative therapy for Duchenne muscular dystrophy. So we're grateful to our partners like Dyne for helping us make these possible, bringing them right into our families' homes and helping us expand our reach across the nation. We welcome Dr. Ash Dugar, Senior Vice President and Global Head of Medical Affairs, and Molly White, Vice President and Global Head of Patient Advocacy, I can't talk today, and KOL engagement. So we're very excited to have them join us. Before we get started, I just want to mention a few things. Today's webinar will be around 20 minutes with a live Q&A at the end of the presentation. This webinar is being recorded, so if for any reason you lose connection, you have to log off, you're not able to make the whole thing, we will have it recorded and we will send it to you guys after the fact, and you can also share that with anyone who wasn't able to make it today as well. So we'll have that on our website and our YouTube for you guys to watch later. So once the presentation ends, like I said, we will have a Q&A. There's a question box, like it's a Q&A box for me, it's at the bottom right. So you can open that and put your questions into there. So if you have any questions throughout the webinar, no matter what time it is, you can just put it in there. We will moderate the chat. So if anything comes through the chat and you guys forget to put it in the Q&A, we will do our best to get to those questions. But for now, I will go ahead and I will hand it over to Ash and the Dyne team for their presentation.
Great. Thank you, Alexa. Hi, I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. And as Alexa said, with me is Molly White, the Global Head of Patient Advocacy. First, I want to thank the JET Foundation for inviting us to this chat to update the Duchenne community on the progress of our Duchenne program, and specifically DYNE-251, which is our clinical candidate designed to treat patients with Duchenne who are amenable to exon 51 skipping. As always, it's a true privilege to speak with the Duchenne community. Next slide, please. These are our forward-looking statements. Please refer to our website if interested in more details. That's all right. Yep, perfect.
Sorry.
No, it's okay. At Dyne, it all starts with our mission, and I know you've heard me talk about this before. We are laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every single day. We're incredibly excited to inform you that we've started dosing participants in our DYNE-251 clinical trial. And our focus today is on sharing an update on our clinical trial progress and details about our trial with DYNE-251. Next slide, please. Great. So we built a robust portfolio focused on rare muscle diseases. Myotonic dystrophy type 1, or DM1, and FSHD represent opportunities to develop first-in-class therapies. And in DMD, we're building a global franchise, starting with our exon 51 program, and we'll follow this with programs for exons 53 and 45 and 44. Next slide, please. So this slide provides an overview of Duchenne, which you all know very well. Our goal is to develop a best-in-class exon skipping treatment targeted to skeletal, respiratory, cardiac, and smooth muscle that greatly increases dystrophin expression and has much less frequent dosing than current agents, with the hope of potentially stopping or reversing disease progression. Next slide, please. Here, I'm going to take a couple of minutes to describe our scientific platform, or the FORCE platform. This platform drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address one major issue in muscle diseases, which is to efficiently, effectively, and reliably deliver antisense oligonucleotides that target the genetic basis of the muscle diseases that we're trying to treat. As you can see, Dyne's FORCE platform consists of three parts: a proprietary antigen binding fragment, or an antibody fragment, or what we call a Fab, which enables targeted delivery of the antisense oligo to muscle, a clinically validated linker, and an antisense oligonucleotide selected to target the genetic basis of disease. On the left part of this slide, you can see a Y-shaped structure, which is a monoclonal antibody. We chose a portion of this, our Fab, as I mentioned, the top piece, as you can see here, because we believe it offers powerful advantages over full monoclonal antibodies, including its smaller size, leading to better muscle penetration, lower protein load, and the potential for enhanced tolerability. The goal is ultimately to enable targeted, effective delivery of therapeutic molecules to skeletal, cardiac, and smooth muscle. On the bottom part of the slide, you can see the linker. We selected the Val-Cit linker for our platform based on its stability and clinically validated safety and efficacy, because this linker is used in products that are on the market right now for other indications. We believe this further de-risks our scientific approach. Now, we're utilizing the same Fab and linker across all our neuromuscular programs, including Duchenne. The versatility and modularity of our scientific platform provides flexibility to deploy different kinds of antisense oligos, ASOs, that are rationally selected to match the target biology for each disease. So where dystrophin restoration is the key, as is the case in Duchenne, it's critical that we select an exon skipping phosphorodiamidate morpholino oligomer, or PMO, that targets the specific mutation in the DMD gene. So in our current study, we're using a PMO that allows for skipping exon 51. Next slide, please. The mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab is engineered to bind very tightly to something called a transferrin receptor 1, or TFR1. TFR1 is highly expressed across the surface of muscle cells, including skeletal muscle, respiratory, cardiac, and smooth muscles. Now, the Fab acts as a kind of key that unlocks the TFR1 and allows our PMO to enter muscles and target the nucleus of the muscle cells so we can address the genetic basis of Duchenne. In short, we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic to muscle with this receptor-mediated mechanism. And by targeting muscle, specifically by using TFR1, we have the potential for a wide therapeutic index, or wide safety margin, resulting in less frequent dosing and also chronic administration. Next slide, please. On this slide, we show our FORCE platform in action in the MDX mouse model, a standard animal model in Duchenne. These MDX mice have a mutation in exon 23. You can see FORCE resulted in robust dystrophin production across key muscles affected by Duchenne with just a single dose at 30 milligrams per kilogram, and we looked at these mice 4 weeks later. The top half of the slide shows some of our data demonstrating robust levels of dystrophin restoration across these muscles. We were able to achieve high levels of dystrophin expression, approximately 50% in the quadriceps, 90% of the diaphragm, and nearly 80% in the heart. We were really excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had significant limitations. On the bottom half of the slide, we show dystrophin localization to the sarcolemma muscle membrane after just that single dose. This localization is important to see to provide confidence in potentially seeing functional benefit. We saw profound results, including about 70% dystrophin positive fibers in the quadriceps and approximately 80% in the diaphragm and heart at 4 weeks. Importantly, our FORCE platform has shown to have durability of effect. If you can go to the next slide, please. Thank you. This slide shows our data on dystrophin-positive fibers in the quadriceps out to 12 weeks. So even out to 8 to 12 weeks, we saw sustained localization of dystrophin to the sarcolemma in the quadriceps, as well as other muscles. And this is really important as we think about the frequency of dosing in the clinical trial. Next slide, please. Next, we evaluated DYNE-251, our clinical candidate, in non-human primates, or NHPs, or monkeys. These studies are a key step in generating preclinical data before going into clinical trials. We measured exon skipping in the quadriceps, heart, and diaphragm. Please note that in monkeys, we cannot look at dystrophin restoration because they are healthy animals. The key message here is that with our FORCE platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. After 5 weekly 30 milligram per kilogram doses, DYNE-251 demonstrated around 50% exon skipping in the diaphragm, 40% in the heart, and about 20% in the quadriceps. Again, really excited to see especially this level of skipping in the heart and diaphragm. We also evaluated DYNE-251 in 5 and 13-week toxicology studies in monkeys. Altogether, we saw that DYNE-251 had a favorable safety profile with no dose-limiting toxicities observed after weekly dosing up to a maximally feasible dose, meaning we dosed monkeys in this safety study at doses much higher than 30 milligrams per kilogram. We're very pleased with the safety profile as we advanced into the clinic. Next slide, please. Thank you. Prior to starting our study, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy, just to name some. Importantly, we've gathered this feedback from across the globe. So key opinion leaders in both the US and outside of the US, including Europe, with experience running in late-stage trials, have provided advice on our overall clinical development program, including our clinical trial design, our patient population, biomarker and functional endpoints, and key safety considerations. And importantly, they've helped us to extract important learnings from natural history studies. It is clear that KOLs believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those from Cure Duchenne and the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. This input has been critical in ensuring a patient-centric design and has included, but is not limited to, key considerations for choosing a trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And based on this extensive feedback, we've developed the clinical study that I'm about to show for DYNE-251 in patients amenable to exon 51 skipping. And here I'll describe the key elements. Next slide, please. So our clinical trial for DYNE-251 is a global, multiple ascending dose, or MAD, placebo-controlled study with a long-term extension. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We're enrolling patients with Duchenne who have mutations amenable to exon 51 skipping therapy. Our goal is to enroll approximately 46 male participants, ages 4 to 16 years, who are either ambulant or non-ambulant. We plan to evaluate the following outcomes in the DELIVER study. These include safety and tolerability, change from baseline in dystrophin protein levels as measured by Western blot, muscle function relevant to both ambulatory and non-ambulatory participants. So that would include assessments to help us understand changes in both lower and upper limb function, as well as lung and heart function. Specifically, we're including the NSAA score and certain time functional tests, as well as FVC % predicted for pulmonary function. And importantly, we will be dosing monthly or less frequently in the clinic. There are 3 periods to this DELIVER trial. The first is the 24-week placebo-controlled multiple ascending dose period. And then, as I mentioned, as patients complete that, they move into the open label extension period for 24 weeks where they receive active treatment, and followed by a long-term extension study period of 96 weeks. With the help of our expert advisors, we're really confident that our study design is robust, will measure key outcomes, and is striving to manage the burden on families. As you can see in the red box here, or the red part of the slide, our DELIVER clinical trial is global and is currently recruiting. We have sites that have been activated in the United States, and we continue to update sites as we activate them on our clinicaltrials.gov listing. And we do expect to have safety, tolerability, and dystrophin data in the second half of next year. Next slide, please. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We're working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices you continue to make. On behalf of Molly and Dyne, I would like to thank you all for your time and attention to listen to this presentation, and we're pleased to take any questions. Thank you.
Awesome. Ash, there is an addendum. Do you want me to go to that or skip to the Q&A piece?
No, I think you can get to the Q&A piece. That's fine.
Perfect. All right. Thank you guys so much. Thank you, Ash, for all of that information. We truly appreciate it. I'm going to stop sharing my screen, but if you guys have any questions outside of what is asked today, or if we aren't able to get to your question, please contact us at Jet Foundation. You can either use our info@jetfoundation.org or my email address, alexa@jetfoundation.org, and we will make sure that those questions get to Molly and Ash and the Dyne team and do our best to get you answers in a timely manner. So we will go ahead and start that Q&A piece. Looks like we have 2 questions. Do you have plans to include adults with Duchenne in future trials?
Yeah, it's a great question. So the answer is yes. So we are, as you would imagine, building out the rest of our clinical development program. And we know that the non-ambulant patient population has been underserved. And so, we will have future trials that go above the age of 16. And those plans will be shared with the community when they're sort of more solidified and ready to be communicated.
Perfect. And then when do you expect preclinical research to be completed for your exon 53 skipping therapy?
Yeah, great question. So clearly, it's notable to point out that we showcased new in vitro data, meaning data in patient cells, for our exon 53 program at World Muscle this year. And so we're continuing to build out those clinical and non-clinical development plans. We're doing the work that you would expect we need to do preclinically to be able to get into the clinic. So as we have more information on data, timing, et cetera, we'll definitely share that with the community. What I would say is that the FORCE platform, because of its modularity, lends itself to also efficient preclinical and clinical development. For the 53 program, for the exon 53 program, the Fab and the linker will remain the same. And so we are going to then interchange the PMO for an exon 51 skipping PMO for an exon 53 skipping PMO. And so much of the program becomes de-risked through the exon 51 program. And so this just makes the efficiency of our non-clinical, our animal studies, and then eventually moving into the clinic, better because we're using the same platform.
Do you have a sorry, do you have a timeline on when you expect in-human DMD trials for that therapy?
Yeah. So right now we aren't, we're still building out our plan. So I don't have a timeline to share at this time. We understand, that we're moving as fast as we can. I can definitely promise you that.
How is it possible to reach out to a third world nation like Nepal and the families living there who could not afford it?
Yeah. Molly, do you want to?
Sure. So we are very interested in including as many eligible families as possible. And we have set up a formal program with our concierge travel service to include families that are cross-border families. So if a family is interested in participating in the study, they can contact sites outside of their country that are open for enrollment, and they can have conversations with those sites about possibly being included for cross-border participation. And if they are accepted, Dyne does have a cross-border policy that supports their ability to do that.
Perfect. Do you have any plans to provide trial program to India or other South Asian countries?
So right now, our plan is to learn from this phase 1/2 study. As I think I mentioned, it includes about 46 participants. So it's it's a relatively small study, and we need a relatively limited number of sites to be able to complete this safety and tolerability study and also look at dystrophin. From there, as I mentioned, we're going to be developing a larger clinical development program that will include more and more sites, of course, as our sample sizes increase for those subsequent studies and then post-marketing studies. So we are definitely committed to having global trials in other parts of the world, like the Asia Pac area for sure. So right now I can't say which specific countries. We're not there yet, but we are definitely committed to opening up sites in Asia, Asia and Pacific region.
Yeah, and I think to your point, Josh, Ash, we're also very interested in inviting families that would like to do a cross-border participation to reach out to sites or to reach out to Dyne and let us know so that we can connect you with active sites.
Wonderful. Is this for deletion 51 or amenable to 51?
Yes. So someone who has a 51 deletion, this treatment would not be appropriate. These are for deletions that are amenable to skipping exon 51. That's right.
Perfect. Well, that is all of the questions that we have. Thank you both again so much for being here, and thank you all of you on the webinar for attending today. Like I said, if you guys, we all think of questions, we sit on them for a second and then think of them after the fact. So if you do have any questions, please reach out to that info email or me directly, and we will do our best to get them to the Dyne team in a timely manner and get those questions answered to you.
And Alexa, we can also share after the meeting, we have a community inquiries at DYNE-TX.com mailbox, and we can share that with you afterwards to provide the families.
Perfect. That would be wonderful.
Absolutely.
Well, thank you again, and have a wonderful day. I hope we see you all on our future webinars. We do have one coming up on November 10th, making driving accessible. So please check out that. And have a wonderful day and a wonderful start to your November. I can't believe it's November already. So thank you guys so much. We appreciate you being here.
Great. Thank you for having us.
Bye-bye.
Bye.